Assalamu alaikum friends. Welcome back to my YouTube channel Microbiology World. I am here with a new topic, another new informative topic about fermentation, erythromycin preparation, about fermentation, introduction, general characteristics of erythromycin. Hope so you like my previous videos and inshallah I am discussing a very informative topic today, that is erythromycin. Now let's start our today topic. Erythromycin, like antibiotics, are also known as macrolides. Macrolides belong to the polyketide class of natural products. Macrolide antibiotics are hydrophobic basic compound. Macrolide antibiotics slow the growth of or sometimes like sensitive by bacteria, reducing the production of important proteins needed by the bacteria to survive. The commonly used macrolides are erythromycin, clarithromycin, roxithromycin and azithromycin. Next, we are studying general characteristics of erythromycin. You can see erythromycin is a macrolide antibiotic that has an antimicrobial spectrum similar to or slightly wider than that of penicillin. Erythromycin was discovered in 1952 by M. Seeger and co-workers from a strain of Streptomyces erythraeus. It is often used for people who have allergy to penicillin. It is poorly soluble in water, bactericidal, particularly at higher concentration. Trade name is emycin or erythromycin. Next, we are discussing general structure. Erythromycin contains three characteristic parts in the molecule. You can see the diagram below. A highly substituted macrocyclic lactone, a glycone. It is a macrocyclic compound containing four-membered lactone ring with the 10 asymmetric centers, a ketone group, and amine desoxysugar glycone, and in some of the macrolides a neutral desoxysugar which are glycosocially attached to the A glycone ring. Glycocyclically attached to the A glycone ring. The two sugars are named L-cladinos and D-dosaamine. Its chemical formula is C37H63 and N1O13. You can see in the diagram below the erythromycin general structure. Next, we are discussing its mechanism of action. Erythromycin plays a bactericidal activity, particularly at higher concentrations. It prevents the growth of bacteria by inhibiting their protein synthesis. Erythromycin binds to the 23S RNA molecule in the 50S ribosomal subunit binding site near the peptidal transferase center, inhibiting the translocation of the peptides transferase center. Thus, binding blocks the exit of the growing peptide, thus inhibiting the translocation of the peptides. Peptide chain elongation is also prevented. Production by fermentation, that is the main point, main thing that we are discussed, how it is prepared by fermentation. Erythromycin is produced from streptomyces and micromonospora. Erythromycin is produced mainly by submerged fermentation, that we have already discussed. During fermentation, different types of erythromycin are produced, like erythromycin A, B, C, and D. The steps for erythromycin production are as follows: inoculum development, Inoculum is prepared from suspension of at least 10 to 8 cells per ml. Sporulation of S. erythrus is done on tryptone agar slant. Cells are harvested from agar plates and suspension is taken in sterile water and stored at 4 degrees Celsius. Next, medium sucrose 5%, corn steep liquor 0.5%, soybean oil meal 1.5%, yeast 1%, NaCl 0.5%, CaCO3 precipitate 0.3%, and pH 7 to 7.2. Fermentation is carried out in a stirred tank fermenter. It should have proper aeration and agitation, pH, temperature, viscosity, and dissolved oxygen concentration. Temperature, fermentation temperature is 33 degrees Celsius. Duration, the length of fermentation is 3 to 7 days. Production of erythromycin production occurs when froth reaches stationary phase. Addition of N-propyl as precursor increases the production of erythromycin. Next, we are discussing the isolation. Mycelium is separated from the broth by filter press. Centrifugation or drum filtration acidic condition helps to separate mycelium. The broth acidic condition is maintained by the addition of butyl acetate, which favors the separation of the mycelium. Also, it dissolves the antibiotic. It is then washed with water. Next, extraction. It is extracted using methyl isobutyl ketone or ethyl acetate. It is then transferred to acidic water. pH is adjusted with HCl, acetic acid, and citric acid. Purification and concentration is carried out in ion exchange resin amber light 50. Elution is carried out by a mixture of organic solvents and water at pH 3 to 8. Erythromycin is obtained as dehydrated salt. The dehydrated crystals of the erythromycin are filtered and dried on a vacuum tray dryer. Clinical uses. Drug of choice in coronobacterial infections, diphtheria, coronobacterial sepsis, erythema, respiratory, neonatal, ocular, or genital chlamydal infections, treatment of community acquired pneumonia, and penicillin substitute in penicillin allergic individuals, prophylaxis against endocarditis during dental procedures in individuals with valvular heart disease. This is the all about erythromycin, its general characteristics, preparation by fermentation, and uses. Inshallah, we will meet in the next video. I request to my lovely friends to subscribe my channel, like my video. Thank you so much.